Hi, my name is Anna. I'm a research associate at Laval University, and here's work I'm doing on predicting antifungal drug resistance in human fungal pathogens. Fungal pathogens can cause life-threatening systemic infections that are difficult to treat because there is a small number of available antifungal drugs and because fungi can rapidly evolve resistance. One of the key strategies in combating fungal systemic infections is quick screening process in the infected patient in order to prescribe the most effective treatment. So far, in Canadian hospitals, this quick screening process is not available for fungal pathogens. To fill this gap, we are developing a workflow that will be easily applicable in hospital settings and will allow efficient and fast classification of the species resistance. The workflow will begin with isolation of DNA material directly from sick patient, sequencing of the subset of marker genes with pre-designed probes, identification of key mutations in marker genes, and application of models to predict species and resistance level from the list of identified mutations. The objective of this study is to use genomes of the fungus Candida albicans. This is a pathogen that naturally occurs in human body, but in rare cases causes invasive candidiasis. We use around 600 genomes of this species as a test data set to identify mutations and genes responsible for fluconazole resistance and test their predictive power. Fluconazole is a drug that targets ergosterol pathway in fungi. It blocks synthesis of the membrane component ergosterol and leads to the production of toxic byproducts. Therefore, enzymes of this pathway are expected to accumulate mutations in resistant strains. We did a genomic scan for fluconazole resistance and identified a set of 190 non-synonymous mutations in 71 genes, including 5 mutations in ERG11 gene, which is a direct target of fluconazole. What was a bit unexpected was that mutations enriched in resistant strains were usually quite rare or constrained to only few resistant strains. On the other hand, there were mutations that were commonly found in both resistant and susceptible strains. In other words, none of the mutations showed strong widespread effect in resistant strains, suggesting that resistance is driven by many mutations potentially located in key regions of the gene, or is driven by synergistic effects of many mutations. Also, predicted variant effects were both neutral or deleterious, suggesting that resistance mutations involve both loss of function and gain of function mutations. Knowing that, we aim to identify the smallest set of variants that can predict fluconazole resistance in Candida albicans. For this purpose, we used a random forest classifier fitted on a balanced subset of a training data and tested its accuracy on the held out data. We fitted models to 190 pre-selected variants selected in the training dataset, and variants located in 11 genes present in a database of antifungal resistance. We also tested models using deleterious or tolerated variants only. Models trained on pre-selected variants performed relatively well in classifying resistant strains, but this was expected because variants were identified in the same dataset. But then models trained on only a subset of genes known to cause resistance were less efficient in distinguishing resistant strains. Training model on a subset of tolerated or deleterious variants marginally changed the performance, with tolerated variants having overall slightly better effect on classification of resistant strains. To sum up, our results suggest that drug resistance in fungal pathogen is driven by different mutations in key regions of many genes rather than a small number of common mutations. Higher predictive power can be reached by increasing number of variants in the model, but what we're trying to also do is to include other types of variations such as structural variation and characterize mutation effects using experimental methods such as deep mutational scanning and computational methods to determine potential mutations that affect protein binding and protein stability, and in effect they can have an impact on resistant phenotype. Thanks for listening, and if you have any questions, please reach out to me at the conference or through email.